The endoplasmic reticulum, ER, is a large, complex, and continuous membrane-bound organelle found in eukaryotic cells. Its name, derived from Greek and Latin words, means little net within the cytoplasm, which accurately describes its labyrinthine structure. The ER's membrane is a single continuous bilayer that encloses a single continuous internal space called the ER lumen or cisternal space, which is separate from the cytosol. The ER membrane is also continuous with the outer membrane of the nuclear envelope. The ER exists in two distinct but interconnected forms, each with its own specific structure and function. Rough endoplasmic reticulum, RER. The RER is characterized by its rough appearance due to the presence of ribosomes on its outer cytosolic surface. Its structure consists of a network of flattened sacs known as cisternae. These stacked, sheet-like structures provide a large surface area for the ribosomes to attach. The RER is particularly prominent in cells that specialize in producing and secreting proteins, such as those in the pancreas producing digestive enzymes or the liver producing serum proteins. Smooth endoplasmic reticulum, SER. The SAR lacks ribosomes, giving it a smooth appearance. It is a network of interconnected tubules. These tubules are typically located more peripherally in the cytoplasm. The SER's structure is more varied and less organized than the RER. Its function is diverse and varies by cell type, but it is primarily involved in lipid synthesis, carbohydrate metabolism, and the detoxification of drugs and poisons. In muscle cells, a specialized form of CR called the sarcoplasmic reticulum stores and releases calcium ions for muscle contraction. The unique shapes of the RER and CR are maintained by a class of proteins called membrane-shaping proteins. These proteins, such as reticulons and DP1, form scaffolds that stabilize the highly curved membranes of the tubular SAR and the edges of the RER cisternae. This structural integrity is crucial for the ER to perform its diverse functions and to interact with other organelles at specific membrane contact sites, where lipids and other molecules. The endoplasmic reticulum, ER, is a crucial organelle found in eukaryotic cells, serving as a dynamic and continuous network of membranes. Its composition and structure are integral to its various functions, including protein and lipid synthesis, calcium storage, and detoxification. Structural components. The ER is composed of three main interconnected structures: cisternae. These are flattened, membrane-enclosed sacs. They are the most prominent structures in the rough endoplasmic reticulum, RER, where they are often stacked and held in place by the cytoskeleton. The outer surface of the RER cisternae is studded with ribosomes, which gives it a rough appearance. Tubules. These are highly branched, smooth-walled, tubular structures. They are the primary component of the smooth endoplasmic reticulum, SAR, and are typically more abundant in cells that specialize in lipid metabolism or detoxification. Vesicles. These are small, spherical or ovoid membrane-bound sacs that bud off from the ER. They are essential for transporting proteins and lipids to other organelles, such as the Golgi apparatus. The ER membrane encloses a single continuous internal space called the lumen or cisternal space. This lumen is distinct from the surrounding cytoplasm and is continuous with the space between the inner and outer nuclear membranes, known as the perinuclear space. Membrane composition. The membrane of the endoplasmic reticulum is a phospholipid bilayer similar to the plasma membrane. It is approximately 50 to 60 A thick and has a fluid mosaic structure. However, the ER membrane's specific lipid and protein composition is unique and crucial for its function. Lipids. The ER membrane contains high levels of specific lipids such as phosphatidylcholine, PT-CHO, and phosphatidylethanolamine, PTD10. These lipids contribute to a looser lipid packing and a more fluid membrane, which facilitates the insertion and transport of newly synthesized proteins and lipids. It has relatively low levels of cholesterol and sphingolipids compared to the plasma membrane. The ER is also the primary site for the synthesis of most of the cell's membrane lipids. Proteins. The ER membrane is rich in a variety of proteins. The RER membrane contains proteins called translocons and riboforins, which are crucial for the attachment of ribosomes and the translocation of newly synthesized proteins into the ER lumen. The SER membrane contains enzymes, such as cytochrome P450, that are involved in lipid synthesis, drug detoxification, and calcium ion sequestration. The two main types of endoplasmic reticulum. The endoplasmic reticulum, ER, is a crucial organelle in eukaryotic cells, and it comes in two distinct forms: the rough endoplasmic reticulum, RER, and the smooth endoplasmic reticulum, CR. These two types differ in both structure and function, working together to carry out a variety of cellular processes. Rough endoplasmic reticulum, RER. The RER is named for its rough, studded appearance, which is caused by the presence of ribosomes on its outer surface. These ribosomes are responsible for synthesizing proteins that are destined for specific locations, such as secretion from the cell, insertion into the cell membrane, or delivery to other organelles like lysosomes. The RER is a network of flattened sacs called cisternae and is especially abundant in cells that produce large amounts of protein, like the pancreatic cells that secrete digestive enzymes. Licensed by Google Smooth Endoplasmic Reticulum, CR. The SIR gets its name from its smooth surface, as it lacks the ribosomes found on the RER. Licensed by Google instead of flattened sacs, the SAR is a network of interconnected tubules. Its functions are diverse and include the synthesis of lipids, fats, the production of steroid hormones, the detoxification of drugs and poisons, and the storage of calcium ions. For example, muscle cells have a specialized form of SAR called the sarcoplasmic reticulum, which stores and releases calcium ions essential for muscle contraction. The CR is more prominent in cells that specialize in these functions, such as liver cells, for detoxification, and cells in the adrenal glands, for steroid hormone production.
Based on the text you provided, here's an explanation of the functions of the endoplasmic reticulum, ER, specifically the rough ER, RER, and smooth ER, SIR. The ER is a key cellular organelle that acts as a workhorse, performing several essential functions. Rough endoplasmic reticulum, RER. The RER is characterized by the presence of ribosomes on its surface, giving it a rough appearance. Its primary roles are protein synthesis and modification. Ribosomes attached to the RER synthesize proteins that are destined for secretion, insertion into membranes, or delivery to other organelles. As these proteins are made, they are threaded into the RER's internal space, called the lumen. Inside the lumen, proteins are folded into their correct 3D shape with the help of chaperone proteins. The RER also modifies these proteins, often by adding carbohydrate groups in a process called glycosylation, protein sorting and transport. The RER acts as a quality control center. Proteins that are properly folded and modified are packaged into transport vesicles and sent to the Golgi apparatus for further processing and distribution throughout the cell. On the other hand, improperly folded proteins are kept in the RER and eventually destroyed, preventing them from causing cellular damage. Smooth endoplasmic reticulum, SR. The SAR lacks ribosomes, giving it a smooth appearance. Its functions are distinct from the RER and include lipid synthesis. The SER is the main site for the production of lipids, including phospholipids, a key component of cell membranes, and cholesterol. Detoxification. In cells like those in the liver, the SER contains special enzymes that make toxic substances and metabolic waste products more water-soluble, allowing the body to excrete them more easily. This process is crucial for detoxification. Calcium storage. The SER helps regulate the amount of calcium ions within the cell. It takes up calcium from the surrounding fluid, cytosol, and releases it when needed for cellular processes. In muscle cells, this specialized SR is called the sarcoplasmic reticulum and is vital for muscle contraction. Steroid hormone production. In certain cells, such as those in the testes and ovaries, the SER is a primary location for synthesizing steroid hormones like testosterone and estrogen.